Alright, so this is my submission for, or this is my video rather, for part A. I'm going to put both parts in the same video. So for the first part, where is that? Okay, so this is kind of have everything kind of like labeled right here of it. And it's written down. So the replication of DNA started from the origin replication site. The DNA, after binding several initial proteins to binding of these proteins for replication, form both strands of DNA, act as a template strands and form the replication fork. Or is it? Just form the replication fork. Single strands of DNA now bind with single strands binding proteins, providing them stability. The helicase, which is right there. Yeah, wait. Yeah. Sorry. I wrote that. Yeah. Oh, you can kind of read it. The helicase enzyme has the activity to hydrolysis, the hydrogen bonds between the strands of DNA, allow the DNA polymerase, sorry for the pronunciation, to access them for the replication process. Now the RNA primers bind to the 3 5 of parental strands. Here, and then now the DNA polymerase 3 starts the elongation of these primers, continuously addition of complementary nucleotide at the 3 end of the new strand of DNA. DNA polymerase 3 only synthesizes the DNA strands in the 5-3 direction. Newly stranded DNA, whose direction is 5-3, is a continuous synthesis called the leading strand, which is used the parental template strand of DNA 3 to 5. The other strands of DNA synthesis also in 5-3 direction, but in the form of fragments called Okazaki fragments, because they use the parent template strands whose direction is 5-3. This strand is called the lagging strand, which is right here. Today, yeah, I did. I did that correctly. Okay, so and then they it's really bad pronunciation. Topomerase. Topomerase is the enzyme that helps to relax the supercoiling of DNA. After cutting the strand of DNA, it moves ahead of the DNA polymerase. Now the RNA primers are adjusted by the RNA enzyme and create the gaps. These gaps is now filling by the DNA polymerase 1. This enzyme binds to the 3 end of strand of DNA and allows addition of complementary nucleotides. Now DNA ligase form phosphodiester bonds. Was that? I'm sorry, I'm just kind of confused what I wrote. Okay. So forming phosphodiester bonds between the fragments of DNA by consuming the ACP form the continuous strand. After completing the process, the replication of DNA consists of one strand of new DNA and one strand parental strand called the semi-conservative replication. And a bit of a mouthful. Took me a bit to wrap my. I think I got most of that right. Correct me, because there's bits stuff on that I'm still confused on. But that was for part A. Let's see. And then for part document for part C. Okay. So for part B, please point out the 5 ends of DNA. There are 2. You can see here. Here it is. I got it all labeled. DNA strands are complementary to each other. So one parent strand will be 5-3 direction from left to right. Another will be 5-3 direction from right to left. As you can see. Further, new strands always synthesize in 5-3 direction. So all new strands will be in the 5-3 direction along the new direction of the arrow. The helic was in part 2. Where would the helicases be located? And for here, we have them right here. Actually, right there, and then right down. Yeah, helicases bind to the lagging strands and move in the 5-3 direction. Yeah, moving in the 5-3 direction along the strand. The position of the helicase is shown. And as you can see, and then for 3, as a new strand leading or lagging strand, please explain how you know this. Please show where the other 3 leading. Okay, so the new strand is a leading strand. Way I kind of went about this one. The template strand, I think this is right. I have a bit of issue with this one. The template strand exposed in the 3 to 5 direction, it will allow new DNA strands to synthesize continuously in the 5-3 direction. This will result in a leading strand. This is the case for the new strand. The template strand that exposed in the 5-3 direction will allow synthesis of shorter strands that will be resulted in lagging strands. As you can see here. There you go. See, lagging strand and then the new strand, leading strand. And there we go.